Morning students, I am Dr. Preeti Ameta, Associate Professor Chemistry, SGG Government College, Paswala. In this video, I tell about the electron transfer iron-sulfur protein, Ferrodoxin and Rubredoxin. Now, see the first slide. In last video, I told you about the cytochromes A, B, C, cytochrome P450 structure and function. Now, these iron-sulfur proteins have some general properties. Both have general properties, ferrodoxin and rubredoxin. They both are non-heme proteins. They participated in the electron transfer redox reaction performed in the mitochondria of the cell. More than one central atom may be present. What is the central atom? Central atom is iron. So the more than one iron atom is present in the any type of protein. Sulfur and cysteine is also present. Next is some iron sulfur protein regulates gene expression. This is just the function of the ferrodoxin, rubredoxin and other iron sulfur proteins. Now two types of proteins are in Yokos, rubredoxin and ferrodoxin. First we take rubredoxin. It has low molecular weight and basic unit of this protein is FeS0. What is the meaning of S0? I will tell you later when I tell you about the structure. It is electron transfer system found in the bacteria. Those bacteria are anaerobic bacteria. Now, non-heme iron proteins. They are also called non-heme iron protein, means NHIP. Next is iron is in +3 oxidation state. Iron found in the +3 oxidation state in rubredoxin. Now FeS distance. What is FeS distance? The bond length of iron and sulfur. It varies from 224 to 233 pm. This is the structure. This is next slide. This is the structure of iron sulfur. This is a tetrahedral type of structure. Iron is present in the central of the tetrahedra. Central metal ion atom is Fe+3 having tetrahedral structure. Iron attached with 4 sulfur groups. They are organic sulfur because they are the part of cysteine ring. And here you see that sulfur are 4. But in the general formula we saw that the FeS0. Now what is the difference? Bond angle between SFES is 104 to 114. This is also vary from 104 to 114. Number of labile sulfur atom is 0. These 4 sulfurs are inert. Why they are inert? Because they are the part of the cysteine ring. They are organic sulfur. They are not free to react with any other type of compounds. These are totally inactive. We can say inactive sulfurs. So they are called S0 in the general formula. These are of two types, FeS0. First is oxyform, another one is reduced form. What is oxyform? Fe is in +3 state. That compound is called oxyform. And if this Fe+3 gain 1 electron, then it converts into the Fe+2, and this is the reduced form of FeS0. Now what is the difference between oxy and reduced form? You look here, you see here that iron +3 state in oxy form and reduced form iron is in +2 state. D5 configuration, T2G3 Eg3. T6 configuration, T2G4 Eg4. This is the configuration of according to the crystal field theory. But if we take ESR, electron spin resonance of these two type of protein, we saw that the ESR active due to the unpaired electron present in the d5 orbital according to the molecular orbital theory. These 5 orbitals have one unpaired electron in molecular orbitals, and in D6 configuration ESR is inactive because 6 orbital is paired in the molecular orbital. So we considered ESR according to the molecular orbital theory, not as the CFT. Here in oxyform ESR is active, so it is paramagnetic due to the one unpaired electron, and it is colored due to the ligand metal charge transfer. This unpaired electron is transferred from the upper vacant orbital, again comes back to the ligand vacant orbital. So this is LMCT, and this LMCT give the color, red color, to the oxyform. And here in the reduced form, the ESR is inactive. Here you can write inactive in place of active. It is diamagnetic because the electrons are paired, and it is colorless because there is no charge transfer in the reduced form. So these are the properties of oxy and reduced form of the Rubredoxin. Now next we take Ferrodoxin. What is the Ferrodoxin? It is the protein having more than one iron central atom present. Basic general formula is Fe2S2. This is called plant ferrodoxins, electron transfer agent, or you can say electron transport agents. High spin iron 3 present in this type of protein. Last is labile sulfur atom, यानी कि ब्रिज एटम जो होता है सल्फर, it is labile. Labile means it react with another. It is very active. So labile sulfur atoms are 2. They are found in the bridge form between the two iron atoms. See at the structure. Both the iron are in the tetrahedral form. 2 iron central atoms. Fe in +3 state. Both the iron in +3 state. It is in two forms again, oxidized form and reduced form. Now see here, both the iron are present, and both the irons are in the tetrahedral form, have 4 bonds, in which 2 bonds are terminal. These are attached with the cysteine ring, and 2 bonds of the sulfur are bridged bonds. They are attached to the another iron metal atom. Now oxidized form, look at the oxidized form. Iron +3 and iron +3 means both the iron present in the structure is +3 state. Both have D5 configuration. Here spin coupling in the ESR, we saw that spin coupling takes place. D5 compounds. Look here, these are the 5 unpaired electrons of the first iron. These are the second 5 electrons which are unpaired of the second iron. These both combine and spin, due to the spin coupling, they form 5 paired electron set. You can say 10 electrons. 5 paired electron set. Number of unpaired electron is 0. There is no unpaired electron. So the ESR is, if the unpaired electron are not present, then the ESR is inactive and the magnetic property is diamagnetic. Now another one is reduced form. Reduced form, gain 1 electron, so only one metal atom, iron, converts to Fe+2 from Fe+3. So both the iron are, first is iron +3, another one is Fe+2. Here D5 configuration, here is D6 configuration. Why D6? One gain of 1 electron. So 5 and 6. In the spin coupling, we saw that molecular orbitals have 5 paired d electron and 1 unpaired electron. This one free electron is active and the ESR active. Due to this ESR active, it behaves as a paramagnetic in character. Now next is second type of ferrodoxin. This is the first type of ferrodoxin which have tetrahedral structure. Second type of ferrodoxin have cubane type structure. General formula is Fe3S4, contains cubane structure. Cubane is derived from the word cube. It looks like a cube. Iron and sulfur are situated at the corners of the cube. Contain cubane structure. 4 labile sulfur atoms. Again, labile sulfur atoms which are active to make another type of bond. So 4 labile sulfur atoms, cube-like structure. Now look at the structure. There is a cube, one upper square and lower square. Upper square have 2 iron atoms which are alternate to each other, and 2 sulfur atoms, they also alternate to each other. And 1 iron atom is at the lower square and having 2 sulfur atoms in the both alternate corners of the cube of the lower square of the cube. Now 3 iron and 4 sulfur present. 4 sulfurs are labiles. Fe and S occupies alternate position of the cube. Iron has tetrahedral structure, so it has 4 bonds. 3 bonds are bonded with sulfur, which are bridged group, and 1 bond of the sulfur of the tetrahedral structure is attached with sulfur of cysteine ring. 3 are bridged and 1 is of cysteine ring sulfur. Bridged sulfur are labile and the sulfur which is the part of the cysteine ring is inert. So 3 sulfurs are labile. Type of 4 labile sulfur and 3 inert sulfur. Overall, if we see the complete structure, 4 labile sulfurs and 3 inert sulfurs. They are of again 2 types, oxiform and reduced form. Oxidized form and reduced form. Fe3, Fe3, Fe3. 3 structure of 3 metal atom, central metal atom. In reduced form again Fe3, Fe3 and Fe2. One is Fe2. The configuration is D5, D5, D5. Configuration is D5, D5, D6. In spin-spin coupling, 2 D5 coupled, but 1 D5 is as such found in the molecular orbitals. To overall 15 electrons, from which 14 become coupled and 1 unpaired electron present. Okay, in reduced form D5, D5 is coupled, and lastly D6 is present. Coupling of 2 D5 orbitals, iron, but 1 is unpaired electron, so unpaired electron present in ES so ES are active and it is paramagnetic. So both the forms are paramagnetic in nature. Now third type of ferrodoxin is again cubane type structure. Fe4, S4, cubane structure, most common and stable structure. 4 labile sulfur atoms which are bridged, 4 non-labile sulfur groups which are the part of the cysteine ring. Look at this cube. Upper square have alternate iron and alternate sulfur. Each iron have 3 bonds with sulfur which are bridged and 1 sulfur of cysteine group. In the lower part of the cube, see the square, again 2 iron which are alternate to each other as well alternate to the upper 2 irons. This is the form of tetrahedral structure, and both the sulfur are another corner of the lower square of the cube. Now these are the complete groups which are present in the iron and sulfur, Fe4 and S4. Oxidized form again and reduced form. These are the 2 forms, Fe3, Fe3, Fe3 and Fe3. D5, 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 D5. Again Fe2, Fe3, Fe3, Fe3 in the reduced form. D6, 5, 5, 5. Here spin coupling, all are coupled with each other, no unpaired electron, ES are inactive and diamagnetic. Here we see 1 unpaired electron, spin is half, ES are active and paramagnetic. These are the 2 forms of the oxidized and reduced form of the Fe4SO. This is all about the ferrodoxin and rubredoxin. Thank you.